Well, good afternoon and welcome to the Iowa Learning Farms Conservation Webinar Series. We're thrilled today to have Maria Lemke, Director of Conservation Science at the Nature Conservancy in Illinois. I'm Liz Ripley, the Conservation and Cover Crop Outreach Specialist for the Iowa Learning Farms, and I am looking forward to learning more about their work at Emiquon Preserve, some science-based restoration and management of functional floodplains along the Illinois River. So at any time, if you have questions, feel free to submit those to me in the chat, and I will turn the virtual floor over to you.
Thank you, Liz, and thank you for inviting me to talk about some of our work here in Illinois. I'm going to start by just including this slide just to provide some of the context of the types of wetland projects that we're working on here in Illinois and the different scales of research. So for a number of years, we've been working conducting research on evaluating the efficiencies and effectiveness of agricultural conservation practices at the farm scale and also at the subwater scale. And the idea is to eventually roll this into kind of a larger watershed scale conservation program. And that's all happening in the Mackinac watershed. We also have these 2030 goals and strategies that include protection and restoration of river floods. So some of our current focus on these floodplain restoration work is revolving around our Emiquon and Merwin Preserve, and these are along the Illinois River here in central Illinois, but also Dogtooth Bend in southern Illinois. So today I'm going to present a high-level overview of our research and monitoring at Emiquon, again, here in central Illinois. So this is kind of an aerial view of Emiquon. In the forefront, you can see the Illinois River. And then in the background here is the preserve site. Historically, this backwater area was connected to the Illinois River. It was separated from the river by levees in the 1920s. You can see the levee here, separating Emiquon from the river. And then in 2016, we completed a water control structure, also shown here on the slide. And this allows water movement and exchange from the river to Emiquon in a managed way. So the first part of the presentation, I'm just going to provide a little brief history of the site of Emiquon, and then I'm going to move into how we assess some of the early restoration efforts prior to having control management through this system here, and then some of the research that we've been conducting more recently that's associated with this water control structure. So prior to isolation from the river, this floodplain area was a system of interconnected lakes and sloughs and wetlands that were lands for 600 generations of Native Americans, including Peoria, the Kickapoo, Potawatomi, Sauk, Fox, and Miami, all who trace some of their history back to this area on the Illinois River. And as you can see here, Thompson Lake and Flag Lake, this is where Emiquon is today. And historically, both of these lakes were connected to the Illinois River. They were in the early 1900s. These backwaters were really some of the most productive areas on the river for both waterfowl and fisheries. And they also provided a lot of this aquatic vegetated habitat, which is missing from the river today for various reasons. So in the early 1920s, like I mentioned, extensive levee construction began and continued until about 50% of the Illinois River floodplain was isolated from the river. And these were primarily done for the creation of these drainage and levee districts for agricultural purposes. So again, this Thompson Lake and drainage area is where Emiquon is today. So the Nature Conservancy acquired Emiquon in 2000. It had been farmed for about 80 years at the time. This aerial shows what the site looked like at that time. This is the historic floodplain in the forefront here, which was in agriculture. There's a lot of infrastructure on the site at the time. Also, there's a lot of concrete infrastructure for cattle. There are some houses on the site, a lot of other types of infrastructure, as well as these agricultural ditches here. You can see that were used to drain the site and keep it dry for farming. In the background, you can see the Illinois River. It flows from left to right on the slide here. And then further back in the background is the U.S. Fish and Wildlife Service, their Chautauqua National Wildlife Refuge. Prior to being isolated from the river, this site has an extraordinary history based in science. And it was intensively studied in the late 1800s and the early 1900s by scientists such as Stephen Forbes and Charles Kofoid. So Stephen Forbes has been acknowledged as the founder of the science of ecology in the US, and he initiated some of the first studies on the Illinois River. Much of them were conducted from this houseboat, this kind of research facility houseboat here shown on the slide. He was the first chief of the Illinois Natural History Survey, and he opened up the Forbes Biological Station in 1894. This research station is located right across the river from Emiquon, and we work with researchers from that station still today. So Charles Kofoid conducted a lot of investigation on these backwater lakes. So Thompson and Flag Lake of Emiquon, you can see his map of these sites from the late 1800s, and he published his work in about 3 volumes or so in the early 1900s. So we have these resources from over 100 years ago of what he was seeing at these sites. So like I mentioned, the Conservancy acquired this site in 2000 and shortly after convened an Emiquon Science Advisory Council, and this lasted for about 5 years or so. The council consisted of scientists and taxonomists and land managers from universities, from federal and state agencies, from other nonprofit conservation organizations. And the goal of this group was to really provide input and guidance for the restoration and management planning before we did anything on the ground. So basically, how do you change this highly agricultural floodplain area back to a floodplain habitat? And there were multiple products that were developed from this. One of these was a hydrologic model to assess the potential sediment accumulation that might happen in Emiquon should we let the river water in at some point. There's another model that was developed to simulate how moist soil would respond to water level management. These are some of the other products that were produced from that working group. So the identification of these conservation targets and really looking at conservation targets that were indicative of the ecological integrity of the floodplain system. And so these are some of the main targets that we're working with today. Ancient fishes like paddlefish, sturgeon, bowfin that need both the river and the floodplain for their life history. Latonia decurrens is it's a floodplain perennial plant. It's endemic to the Illinois River Valley, and it really is an indicator of a functional floodplain habitat. Other conservation targets include waterfowl and wading birds, freshwater mussels, and then these backwater floodplain plant communities that again are really missing from the Illinois River Valley today. So to assess the effectiveness of restoration efforts at Emiquon, this framework was introduced as part of the process, and it includes the development of these key ecological attributes. We call them KEAs, and then indicators for those KEAs. So this was completed in 2006, and we still use this framework today to assess how the restoration is working. And I'll just mention too that this framework, this KEA framework, was modeled after a publication by Parrish et al. In 2003. If you're ever interested in looking into that more in depth. So since 2007, actually, biologists from the Forbes Biological Station and the Illinois River Biological Station have been monitoring many of the KEAs for us, focused around waterfowl, fish, and moist soil and aquatic plant communities. And there's a lot of other research happening as well. For instance, there's a lot of research around fish feeding and marsh bird use of different habitats. We've conducted a lot of work with the University of Illinois Springfield on invertebrate and microbial communities and how they're responding to restoration and the prairie habitats as well. So there's a lot of research happening out here. But what I'm going to focus on today is specifically these KEAs and how we're using those to assess the restoration in particular. All right, so this is just a subset of some of the KEAs that were developed for fish, riverine and backwater fish communities. Just to show an example of how we track these KEAs. So for example, the KEA in this case is the fish community assemblages and composition. And then the indicators focus on the native fish species. So the desired ranges include, how many native fish species might be out there, how much of the biomass out there is made up of native species, and then the catch rate of particular fish species. So data from the KEA monitoring is input annually into a spreadsheet like this one. And each of these cells include, some basic information on the data. It also includes citations of where you can find the full report for that sampling season. And then they're also color coded. So red means, they're not within the desired range. Green means we've reached that desired range. And you can see that if I just compare here 2008 to 2020, that early on in the restoration, we were not reaching that desired range of native species numbers. And eventually, as of 2020, we did reach that 25 to 30 native species. So this is a really good way of tracking the different aspects of our conservation targets over time. And you can see that there's a lot of ways we can track this over time, if they moved from red to green, if they're moving into that desired range, or if they're going the opposite way and moving out of that desired range. And it helps us assess that restoration. All in all, the council identified 26. We have 26 of these KEAs, and we have 60 different indicators, many of which are being monitored and tracked during the restoration. All right, so the next few slides are going to just show some examples of rolled up the data for these KEAs. As part of this, we did a big assessment back in 2014, a collective assessment across the board to see kind of how things were moving. And this was prior, again, to the construction of that water control structure. So Parrish et al. States that in order for any target to be considered conserved, that all the attributes have to be, for that target, have to fall within acceptable ranges of variations. They have to fall within those indicator ranges. And if we just look at moist soil vegetation from early restoration to 2014, we can see that overall the moist soil vegetation did really well. In fact, it reached that conserved status in 2013, meaning that it reached that all the indicators were in that range. So if we look at the fish, they did really well too. Actually, a lot of the fish were stocked in 2008. And overall, those communities, we could track those communities over time, and they did very well. And they actually came close to reaching conservation status in 2013, and had some points to do with, spawning, increased in spawning, and nursery habitat at this site. And as we look at what's happening in 2013, this increase in conservation status coincided with an historic flood in 2013 when the levees overtopped. The Illinois River flowed into the site over the levees for the first time since they'd been constructed. And so what we found after this historic flood was we started collecting for the first time riverine fish species. So we collected fish adults and the larval forms of these riverine species that we hadn't seen before at Emiquon prior to the flood. And so what this also really indicates is how important it is for the connection of these backwater areas to the main stem of the river and that connectivity. So the conservation status of the aquatic vegetation has really varied considerably over time. Ultimately, we saw a decline early on. That it did really well in the restoration, and then we saw this big decline from about about 83% of indicators being in range to about 17% being in range. And if we look at other water quality parameters, we see that this decline between 2009 and 2010 really coincides with this change in transparency between 2009 and 2010. So what happens is we have water transparency. Early on, it was crystal clear water, and the water transparency was about 1.2 meters deep. A big change happened between 2009 and 10, and since then, it's been the transparency was really reduced to about 0.4 meters. And that really coincided with a lot of changes in the aquatic vegetation as well. Even so, I'll just note that as you look at Emiquon, if you ever visit Emiquon and look at across Emiquon, there's a lot of aquatic vegetation out there, and it's great. It provides critical habitat, that then its backwater areas that is very much used by waterfowl and other species. So just to kind of wrap up that early assessment, in terms of the KEA framework, it really did provide the basis for developing our strategic and systematic long-term monitoring program that we've been conducting since about 2007, 2008. And that timely assessments of these data provide, this monitoring data gives us timely assessments. We get them annually every year. And it also provides our long-term, looking at long-term trends of conservation targets and what their status is across time. And finally, we're using this long-term data to look at the potential roles that water level management and connecting the site back to the river under managed conditions might do. So we're looking at, and we constantly kind of look at these indicators and refine them as more data becomes available. We're using this data to develop and test hypotheses associated with water level management. So how do we reduce potential threats by, bringing water in or letting water out? How do we do that and still achieve restoration goals? And looking into the future of how to make management decisions at the site. So if you're interested in, this is a really, really high overview. And if you're interested in knowing more about these responses, these different targets to restoration, about responses of the site to the 2013 flood, and as well as other papers, these were all published in a collective special edition of Hydrobiologia several years ago. And many of these are open access. So if you want to know and find out more, you can find that online. So, like I said, when the Conservancy purchased this site in 2000, one of the goals was to restore these wetlands and lakes and the forests and prairies associated with the site. But another big goal was to restore the natural hydrology that resembled the historical flood pulse regime when the floodplain was still connected to the river. And so the last part of this presentation is just going to cover some of the early research we're doing around this managed connection that was completed in 2016, and that we can use to exchange water between the floodplain and the river. This is a close-up of the gate of that control structure from looking at it from the river side. And the control structure is really 2 8x8 culverts that go through the levee. And these gates are set up with slots along here so we can put in nets for sampling fish. We can put in, stop lives and other monitoring equipment. So we're monitoring kind of water, the flow coming in and out, and fish movement through here. And while there are multiple considerations and some concerns related to connectivity, we do have some specific KEAs that are associated with this managed connection. So for instance, managed connection would provide backwater habitat and nursery for riverine fishes. It provides some of the hydrological requirements for vegetation, like frequency of flooding and timing and duration of recession. It provides moist soil habitat for migratory waterfowl, as well as foraging and nesting habitat for other breeding birds like rails and coots. And for the last few years, we've been collecting information on what is being delivered from Emiquon to the river. So these first KEAs really focus on the health of the floodplain. This last one that we're really focused on now is how is the floodplain benefiting the river? What, as we let water out of the floodplain into the river, what are the benefits that it's supplying to the river system? So we're looking at, we've been measuring kind of what we're calling the secondary production that's delivered from Emiquon to the river for the last few years. So just real quickly, while primary production then relates to the formation of mass for those organisms that can produce their own food, like plants, the secondary production is actually the formation of biomass of those heterotrophs over time. So those organisms that do not make their own food. So we've done a lot of research around aquatic invertebrates, the secondary producers that live in the system prior to restoration, during restoration, and continued research on that, just to monitor their responses to restoration over time. So we have a lot of background information on the invertebrates that live in the sediments, that are emerging out of the system, the invertebrates that live on the plants, and the planktonic invertebrates, those that live in the water column. And those are mostly made up of zooplankton. And this is what we're focusing on now in terms of when we're measuring what's being exported out of the system in terms of secondary production. We're really looking at what's the plankton, since they're in the water column, what's being exported in terms of the zooplankton out into the river. So this just gives you an overview of the hydrograph showing water surface elevation through time from about 2003 through 2021. The water control structure was completed in 2016, which is right about here. And then we've had several different drawdowns since then, some using gravity flow, which are depicted by the red circles, and then some using electric pumps, which are depicted by these green circles. These squares just show some of our previous invertebrate sampling through the years. And then what I'm going to talk about today is what's being exported out of the system, particularly in this 2020 year where we had a really long drawdown. So this just shows kind of what our sampling setup looked like. So here's Thompson Lake and Flag Lake. This red circle is where the pumps are. And then these yellow circles are our sampled water coming directly out of Emiquon to the river. Then we continue to sample down this pump house ditch and into Thompson Lake just to get some context of what's being produced in the system versus what's leaving the system. And this is basically how we sampled. This is 2020. We have a hand diaphragm pump with a weighted hose. And so we can lower and raise that hose throughout the water column to collect samples throughout the entire water column. And then we had a lot of field measurements that we collected as well during all this. Mostly what I'm focused on is just the zooplankton piece of this, given the time. So again, what were we collecting when I say zooplankton? I don't know how many people know what zooplankton are, but they're a really diverse group of animals that live in the water column. They're mostly microscopic. Some of the larger ones are shown here. You can see some of these without a microscope, generally swimming around. So the cladocerans at the top here generally have this really large eye spot, these swimming appendages that stick out. They're transparent. Carapaces, you can see the eggs developing in these particular individuals. And then when I talk about copepods, they're kind of torpedo-shaped animals. They carry their eggs externally. This little alien-looking creature is called an oplei. It's the larval form of a copepod. And ostracods, they look like little seeds a lot of times, like little clams in a way, very tiny, and called seed shrimps. And the other microscopic group I'm going to talk a little bit about are rotifers. They're one of the smallest multicellular animals that exist on the planet. They're fascinating, quite diverse. And when you look at them really close up, you can see some of the actual specialized organelles within these little animals. And finally, we did do some algae collection. We looked at algal biomass leaving the site, and again, I'm going to focus mostly on the zooplankton for today. All right, so the next two slides just show some of these high-level results of the export in 2020. So we saw the highest, this is abundances, so number per liter being exported out of Emiquon into the river. You see the highest numbers really occurred in June, between about 4,200 and 13,000 individuals per liter of water that was exiting the site. And then it really just dropped down and leveled out to about 2,300 individuals per liter the rest of the summer. If we break this down to those three major groups I talked about, you see that most of the individuals leaving, the abundances were mostly made up of rotifers, about 70 to 90% on any given sampling day were those very small rotifers. although you do see a few cladocerans and copepods throughout the year, mostly cladocerans mostly early on. This line just shows biomass. So here's the biomass leaving. And again, it really follows that trend of abundances. So really high biomasses early on in the drawdown, and then leveling out the rest of the summer. If we just look at biomass, you have to see a really different picture. So early on, a lot of this biomass leaving the system were those large-bodied zooplankton, mostly cladocerans. And these really, it just follows their life cycle. They usually are most abundant in the spring and start dropping off early summer. So this is really reflecting that cycle. And then it switches over to rotifers and copepods. So still a lot of the biomass is made up of these larger bodied individuals, which is really good for filter feeding fish that depends on these zooplankton for feeding. So in general, you know, I didn't show a lot of the data from the river, but the potential and these systems to contribute algae and zoomass to the river is really huge. The large river systems tend to have really much lower algal and zooplankton biomass in the system, and they really depend on these backwater systems to provide that for filter feeding fish. The export from Emiquon, we got some real numbers on that, you know, 1,300 to 13,000 per liter, which may not sound like a lot, but if you look at how many liters we pumped out of there during that drawdown, about 17 billion liters, that's a lot of organisms that were contributing from this backwater, really productive area to the river system. And the algae too, I didn't show you the data from that, but about 10 times more algae was exported into the river than is in the river. That's really important for things like the zooplankton in the river and also for the mussels. The zooplankton are really important. You know, the fact that we're being able to contribute plankton to the river is really important for these native fish, the filter feeding fish, like especially paddlefish. You know, you may have heard this introduction of these non-native carp to the system, silver carp and bighead carp especially, and they are voracious filter feeders, and they filter zooplankton as well. So they're competing with our native fish like paddlefish for the zooplankton. So being able to support export into the system is really important for the native fish species. So in general, yeah, these drawdowns are really important to, you know, kind of providing that historical function of providing food to the system, as well as drawing down the system for the moist soil production, for waterfowl feeding and such. So creating a natural hydrologic regime is still, you know, we're still learning, but we're starting to get more and more data on that. So I'm going to stop there and take questions and move to this next slide on certified crop advisor information for Liz.
Thank you so much. Yes, and if you have questions, feel free to submit those in the chat. If you joined us today to earn a CCA CEU, please provide your name and CCA number to Alana Whitaker at alenaw@iastate.edu by 5 p.m. today, and we'll get that credit submitted on your behalf. And if you want to go ahead and click to the next one for me.
Yep.
We have a QR code here for a voluntary demographic survey. If you filled it out for a previous webinar already this year, thank you. You do not have to do it again. We only need one response there. So if you want to go one more time, you can join us next week as we dive into monarch butterfly biology, ecology, and conservation needs with a recent Iowa State graduate, Kelsey Fisher, who's now at Connecticut. So join us then. And we do have some questions that have come in. So they're curious on the fish side of things. Have you done any gut analysis to see what kind of predation pressure from non-native game fish on your native fish within the preserve?
So predation from non-native fish on native fish. You know, we have almost the only non-native fish I can think of right now are mostly like the filter feeding carp, bighead. We had some bighead and silver carp that came in through that flood in 2013. There's common carp in there as well. But I can't really think of a non-native predator that we have in there. We have stocked some alligator gar pretty recently through the Department of Natural Resources. There are some big predators in there, but they're native species. And I do believe they've done some work on looking at gar, some of the alligator gar and their feeding habits in the system.
Forgot I was muted. Sorry about that. Note that maybe largemouth bass is considered non-native.
Largemouth bass is native, and we've stocked a lot of largemouth bass in there, and there have been multiple studies on their feeding habits as well. Yeah, and a couple of master's projects actually have worked on largemouth bass feeding. Yeah.
So you mentioned invasive carp in terms of helping feed other fish besides them, but have you noticed any issues with invasive carp in the preserve, especially before and after the levee break or flooding that occurred?
Yeah, so common carp were always in the system and are still in the system. And, we haven't seen a large scale effects of common carp out there yet, although we know that the potential is there. They can root up, they root along the bottom and uproot vegetation and create increased turbidity, which then continues to reduce aquatic vegetation. So we're monitoring that very closely. So the Illinois River Biological Station does the fish monitoring very closely. We're tracking the densities and biomass and abundances of common carp, as well as the other carp species. Silver carp and bighead did come in. We could see them coming in during the flood, so we know they're there. And but apparently, what we're seeing is they're not the silver carp bigheads are not reproducing out there. It's the science says that they can't reproduce in these backwater areas, and we're not seeing any evidence of their reproducing out there. They're getting big, but they're not reproducing. So it's just it's part of the system. So we are definitely tracking that and thinking about the future and if we need to take action or not.
Yeah, so along those invasive species, if you want to maybe flip back in your slides, there's a question about the physical structure that was constructed, and if there's any screens to prevent invasive species.
Yes. So let me flip back to the gate. Probably this picture.
We're seeing your desktop right now, unfortunately.
That's okay. I stopped it, so
There we go. Okay. Yes.
All right, and then I'll flip. Okay. So this is the control gate system, and we do have we put in big. Actually, we've only let water into Emiquon once, and it was very brief. And so we've mostly let water out, but we do have big screens in here to keep fish from moving, the bigger fish from moving into Emiquon. So we're not letting big carp, come into the site through the gate. Now, we also, one of the reasons we did let water in very briefly was because we also sample for fish. So when we were letting water in, we take a sample and look at the fish. And if we see young of year carp coming in, we'll shut it down, which is what we did. So we can time it also when we know that they're not reproducing out there. So we don't have a lot of little fish out there, a lot of young, carp in the river system. So we can time it through the season, but also we can look and catch it when we try to let water in once. So.
And then maybe flip to the slide. They were curious about that publication name that you put up there one more time.
Sure.
While you're flipping to that one, curious here, someone wants to know, is public hunting access to the Nature Conservancy land being considered in Iowa and or Illinois?
I can't answer for Iowa, but in Illinois, at Emiquon anyway, we do waterfowl hunting, and there's an online application for that. I think I don't really work with that mostly, but you can sign up for a blind, and I think they pick people for blinds randomly or something. So there is access for that. It's free. There's also There is a deer hunting out there, but it's kind of a different system. It's not public. And there's also public fishing, though, which is also free. But for fishing, we do require a permit for all of that, so we can track, how many people are fishing and things like that. And there's no only electric boats, so no gas powered motor boats out there. So there is access, and there's a really great use, like walkway and interpretation trails and things like that out there as well. So there's quite a bit of public access.
Question here about nitrate measurements that were done. And did the algae fluctuate in response to nitrates?
Undoubtedly, yeah. We do see algal blooms pretty intense, pretty early a lot of times. And I'm sure they're responding to phosphorus release from the system and nitrogen and phosphorus cycling out there. We don't see really high nitrates in the system in the backwater areas. We did do a big flush, like right after when we first restored the site. We drained a lot of water out and then let it fill back up. And I think a lot of that residual nitrate from the farming and stuff probably exited at that time. We see a lot of cycling out there. There's a lot of plants and a lot of ways where those nutrients are being cycled in the system in a lot of plant biomass. So it does move through the system, but there are times when we do see pretty high algal blooms, yeah, and blue greens as well.
So this person asks what the public response was to this restoration project. Were there any strong reactions, either positively or negatively?
Yeah. Again, I'm talking from what I have heard, and I wasn't with the direct, recipient of public response. But for the most part, I think people have been really, really excited about it. And I've heard, people use the site, very excited about the site. There were a few, there was always some doubts, and there were early on, especially about, taking farmland and putting it back into floodplain. But for the most part, a lot of people in the area remember when it was still floodplain, or their fathers talk about it was still floodplain. They remember the stories. They were pretty excited. It's been a really good positive response overall, I would say.
Excellent. I'll give one last call if anyone has any final questions. But with that, if there's any final thoughts or comments you would like to make, Maria, we appreciate you coming on today.
Thank you. I don't have any final comments. But if there's any other questions, I'd be happy to, if things come up later, just let me know.
For sure. Yes. Well, thank you again.